And we're live. Welcome to this webinar from the series KNIME101. So today it's chemistry for beginners. My name is Alice. I'm a data scientist in the education team here at KNIME. And actually I did my PhD in toxicology. And chemistry for beginners is something that would have come really handy in for me back in the day when I was still in the wet lab. Because if I would have known how easy it is to calculate descriptors of molecules, that would have made my life much, much easier. And with that, I would want to welcome our speaker today. I'm super happy that he's here, Hiroki. He's coming from Japan, so actually it's really quite late for him already. So Hiroki is in charge of chemistry at X-Force Therapeutics. So they're a company that are doing drug discovery for RNA-targeted small molecules. And he's also a ninja. So in 23, he participated in this competition, our KNIME competition. He also flew over from Kyoto, I think, to meet us here in Berlin. And that's really, really cool. And I do think, Hiroki, I think you have a background in medicinal chemistry, right? So maybe you also want to introduce yourself a little bit.
Okay, well, nice to meet you, and it's great to meet you. I am Hiroki Yoshida, living in Kyoto, Japan. And it's very actually honored to have a presentation today. I didn't expect that so many people join this webinar since it's my first time to talk in KNIME. And also I'm so excited, but at the same time, I'm feeling kind of weird because I was an audience for this type of the event before. And now I'm sitting completely in opposite position. It's kind of
I feel you. We're actually all scientists and not doing something. We're not the pros, but I think we're doing good.
But this is actually the good opportunity for me to kind of talk about this because the KNIME is really useful. And as Alice introduced, my major was chemical biology at the university, and now I'm doing the medicinal chemistry and the cheminformatics. And I personally actually had some connections to Germany. I mean, I was invited last year as a K-Ninja things, and I enjoyed it a lot. And I studied actually the pharmacy at the Heidelberg University, so I have some connections. And since then, Germany is kind of my second country. So that even for honeymoon, I have been to Germany.
That's super cool. And we also just discovered actually we have the connection because I studied in Heidelberg. I just came to Constance to do my PhD.
Oh.
And with that, I think, okay, we want to get started. I'm really, maybe the others are as well as me, excited to learn about the descriptors. That was good for me.
Why not? Yeah, let's get started. Is it screen sharing now? Yeah. So, well, anyway, today's talk is a KNIME in chemistry for beginners. And this will provide you how you can analyze the chemistry in KNIME. And I hope that you can start to use KNIME after this webinar. I'm sure I'm from Japan, so I'm now kind of it's evening. But maybe most of you are in Europe and it's still kind of in the morning, early morning. So after this webinar, you can maybe start to use KNIME for chemistry. And let's get started. So my presentation have 4 topics today. At first, I will explain the current situation, like chemistry in data science, and why KNIME for chemistry, what can KNIME do, and how to run KNIME easily. So the current situation in chemistry, sorry for kind of busy slides, but it needs, we need some explanation for the background. So the AI tech such as ChatGPT are rapidly gaining attention across a wide range of fields, and I'm pretty sure that you have heard such kind of IT tool like every day. And as a result, the knowledge and the skills in IT or AI are no longer limited to the specialist. And they are becoming essential. Wait, can I just make it the later point? Yeah, it's visible now. And they're becoming essential tools for anyone doing anything. And I recently see many programming schools, at least in Japan, even for kids, like 10 years old kid can start to learn programming. Who could imagine this type of situation 20 years ago, actually? It was kind of very surprising. And in drug discovery field, especially the integration of the programming tool is becoming increasingly necessary. Recently, AI-driven approaches such as AlphaFold for protein structure prediction, molecular dynamics simulation, predictive automated model, and as many other IT tools have gained significant attention. These technologies are opening the new possibility for innovation and speeding up the kind of path from discovery to development. But this makes the situation completely different. Let's talk about chemistry today, even kind of wet lab researchers are expected to have kind of fundamental understanding of chemistry in programming now, and the conducting laboratory experiments alone is no longer enough. So, well, how should wet lab researchers start to learn programming in general? Well, just code it. Let me just code it like this. And this is actually the issue because it is so difficult to learn coding from scratch. I would say that Python is quite useful since we can do anything about chemistry. Many dry lab researchers in chemical field use Python as a standard programming. So it has a lot of library as a ready-to-use status. So if you do not mind reading a lot of words like this and thinking logically with text and blocks, I would recommend running Python instead of KNIME. However, in many cases, for researchers who have mainly focused on the wet lab experiment, handling chemical structures through the Python like this can feel overwhelming. So just seeing the huge block of code and text can discourage it. Then the KNIME is one solution for it. This is actually the simple KNIME workflow. You can just perform almost everything you need simply through the drag and drop operations without writing and reading code. I mean, we can actually put some Python code in kind of the middle of the workflow in the KNIME. So this is so-called low-code programming, but it's still useful. And the visualization is also quite easy in KNIME. So I'm pretty sure that all chemists feel comfortable to see the structural information during the coding. And this is all for explanation of current situation, coding in chemistry.
Yeah, I think a lot of people can relate to the situation that, yeah, actually we're supposed to know the stuff about the molecules, but we are not good enough in the coding. I mean, that was the case for me. So do you actually have some examples for us, like actual workflows? Something to show?
Course. Yeah, I mean, it took a little bit longer to explain the current situation, but yeah, I will just show you some concrete examples. So why KNIME for chemistry? I put the 3 points here. So it's quick to build up the programming, and it's also interactive to visualize chemical structures. It's also easy to develop useful applications. Let's dive in the first one. So here I will emphasize how quick you can build the programming in KNIME. This is actual workflow to generate the chemical descriptors from structural information. It's just 3 steps to make it. The first, drag and drop the input file from the folder to the KNIME, like maybe SDF file is the easiest way. And the second, drag and drop the appropriate node. Node is here. So third, it just connect and run. But in case you have never touched KNIME, I'll just give you kind of brief introduction. I mean, this is called node. So node is the square one, and this is a library of the node. And the node is connected by arrow, the black line here. And all these nodes are called workflow. So this set is called workflow, and each node has some functions. For example, the first node here is to read the SDF file. So SDF file is a standard file which you can download from chemical company. Like if you want to buy the chemical library from the company, then you will get SDF file. And it contains a lot of compound information. And the second node here, it's desalting. So sometimes the compound is purified with salt, like hydrogen chloride and HCl. Because the compound is protected by the bulk, and you need to deprotect it. And after this, the salt is still there, but we need to remove it to calculate the proper property. That's why we need this node. In third node, it generates the chemical descriptors based on structure. And in fourth node, I just change the format, which makes us possible to save it as a CSV file. And the final node, it just saves the data as a CSV. So furthermore, you can just click third node here. Just click it, and it's selected actually. And at the bottom, you can see the chemical structure. So it must be very helpful to understand the programming, because at least I feel very comfortable to see the structural information during the programming when I started to learn it. And the second reason, KNIME has an interactive function. So what's interactive? This means that you can just choose what you want to see. Maybe it's a little bit difficult to understand, so I'll just show you some detail here. this is the actual workflow, and these 5 nodes are shown in the previous slide already. And I just put one node here. And in this node, there are several nodes here. It's just a viewer. And let's say you downloaded the chemical library data from some company, and then you want to choose the compound having these properties, like log P should be between 4 to 5, TPSA should be 50 to 70, molecular weight should be 340 to 360. Then you can just do it interactively. So you can, each bar has meaning. So this is a S log P, and the TPSA, and molecular weight, number of the hydrogen bond donor, acceptor, ring, aromatic ring, and the FSP3. You can just drag what you want, like a 4 to 5 in this case. And the TPSA should be like a, what's this, 50 to 70, and the molecular weight should be 340 to 360. Then these 5 compounds are selected. So I just show you a quick example using the actual line, maybe. Can I just, maybe you see my screen, right? So that's the folder. Yeah, right. And if you have an SDF file in folder, you can just drag and drop it, and it automatically, automatically node is installed here. And you can just click here to see actual structure. And in this case, this SDF file contains like a 10,000, over 10,000 compound, and structure information is here. And this is actual view of such. I'll just make it bigger. If you want to search, for example, log P should be 2 to 4, TPSA should be less than 100, let's say. The molecular weight should be kind of very small here. And the hydrogen bonding donor should be 3. Then these compounds are selected. It's so easy to select the compound with some specific property. How can you go back to the slide? Yeah, it's fine.
Maybe also just as a quick, what Hiroki is showing here now, the gray thing, that's actually a component. So this interactive view, that cool thing that he's selecting something and it's being propagated. For the ones who don't know that, we also have a webinar where we introduce how to build them. I think it's on YouTube, just to sprinkle it in to avoid confusion.
Thank you for the explanation. Sorry, I just kind of skipped it. And this is for chemistry for beginners, so I mainly focus on kind of chemistry.
That's true. That's more KNIME and not the chemistry bits, right? Yeah.
Well, anyway, you see my screen now, right? Yeah.
Yes.
So it's also very easy to develop the, for example, this is a reagent storage system. And usually the substructure search by Python is a bit troublesome, but it's so important to manage chemical reagent in general. I'm sure that some of you are working in the synthetic lab at the university, and many people come and leave in each year. It's so hard to control reagents in the lab. A rich lab still can buy the commercially available software for reagent storage system, but it's quite rare case. But you can just make it by KNIME, like freely and not so difficult. It's so simple like this. This is, wait, make it laser point. Yeah. So this is the actual workflow. And the red box here is for registration. And the blue box here is for deleting reagent that you have. And the green box is for substructure search. And the reagent name, I mean, it is also possible to make this type of the system using the Excel, but there are 2 difficult points. So reagent name and substructure search. The reagent name is not always identical in chemistry. I mean, some people call this way and other people call that way or something like that. The substructure search is also necessary for storage system. I mean, you will probably search the compound based on the structure. So this workflow actually solves both problems. You just need a CAS number instead of the compound name as an input when you register it. And it automatically fills the compound name, so there's no name difference between people. And the substructure search must be very important for organic synthesis. You do not need to buy reagent you already have in your lab, and you can just draw it like a chem draw. Yeah, 1 point. So sometimes if you use a node, you will see the words deprecated above the node. But it means that this is the old version of the node. So there is a newer version, but you can still use the deprecated node. And anyway, that's all for introduction of KNIME for chemistry at first.
Cool, that's really nice to see. Just to cover it again, so there is this one node that does the descriptor calculation, right? You drag and drop that from the SDF, so we're importing the molecules, and then there's just one node. So you actually, what you showed there with the individual bars, that's descriptors that you're calculating just in one node. Is that nodes that you're using there, are they actually free, or like can you maybe cover a little bit about these libraries or the functionality of them?
I think like many of people have heard about kind of RDKit or some kind of a very famous library for chemistry in programming, and actually people can still use it in KNIME too. I mean, these nodes are kind of native node for KNIME, but I mean, the RDKit or other Vernalis is also possible. So then I will just show you how to extract the chemical descriptor using the RDKit, and also how to extract the 3D structure or PDB or something like that. And I share more other application by KNIME. But before we jump into the case study, I'd like to quickly go over some basic concept of chemistry in data science. So when working with the chemical structures, we cannot directly handle them as it is in code. You can see my screen. Yeah. Instead, we need to convert them into the machine readable format like this. I mean, these are kind of difficult for me, at least, and for human beings, it's probably difficult to read it, but for machine, it's easy to see. I mean, this all line means the same compound actually. But one of the most well-known way to represent the molecule is using smiles, this one. Honestly, before diving into the chemical data science, I highly recommend spending some time learning a grammar of smiles, this one, because it's extremely useful when you face the error. And the other commonly used format includes inch key, smart, and fingerprint, and each has kind of different properties. And actually, there are several variations of the smiles and fingerprint or something like that. It's a little bit complicated, but for simplicity, I won't go into them today. And if your compound requires a 3D structure, you can just convert it into the model file like this, which is often stored with.
Woki, do you have a chance to kind of hide the toolbar, because I think we can't see the last point on your slide?
What do you mean? I mean the icons of the map.
Yeah, exactly.
But how?
Because right now it's smiles and SDF. Like I can only guess what the last line is.
Okay, I see. But I don't actually know how to change it though.
If not, no worries. Like we'll cover it. You can also just.
Okay, it's fine now.
There we go. Cool. Thank you.
Right. Yeah. Where was I? Oh yeah, the last thing. So the 3D conformation. I mean, if your component has a 3D conformation and you want to save it, then just use a model file here, which is often stored within the SDF file. And the SDF file includes the model structure along with the metadata, like a compound name and other properties. Or to summarize, when doing the data science with chemical information, it is important to choose the right format for your work. But today, I'll just show you some simple and practical example using KNIME, and SDF and smiles are probably the first step to dive in the coding in chemistry. So the first example is to generate the chemical descriptors. I mean, it's quite similar to what I showed in the first slide. But anyway, the chemical descriptors are like TPSA or molecular weight and log P and so on. You can just start to draw the structure maybe by ChemDraw. It is also possible to draw structure in KNIME, but I believe you're more familiar with the ChemDraw. So please draw anything you want, and then just open as a quick file and save it as the SDF file. It's easier to start with something you already know, right? And but if you can actually see the chemical property in ChemDraw too, but you don't want to like check 1,000 or 100 compounds in ChemDraw, so it's useful for that kind of large data things. The second step is to import the SDF file, which made from the ChemDraw, And this is actually the SK file, and you can just drag and drop it. And please choose the RDKit descriptor calculation node from here. It is not a native, like a usual normal node, so you have to download it after downloading the KNIME itself, but it's still useful. And actually, that's it. And connect and run. So your descriptor is going to be here. And the next is about Vernalis. There are some more advanced technology like this. And I'm actually connect the PDB and get the crystal information. If you have many data, then it's going to be very useful. Like you can just extract the ligand information from PDB. And some of you may hear about the PMI analysis. Sorry, the figure is a little bit too small, but it categorizes the compounds based on the rod sphere disk in terms of the structure. This is also very easy to do it in KNIME. And the FAST file is used for DNA or RNA. KNIME is useful not only for chemistry, but also for biology. As you know, we're getting many knowledge from kind of different fields. It's important for research. So you can just use a KNIME as a tool to research both chemistry and biology. And the last one is the MMP analysis, like a matched molecular pair analysis. And this is also very useful for SAR study in drug discovery field. But before next page, I will just introduce. So this is like a traffic signal. If it's green, it just goes well. But if it's red here like this, it stopped here. I mean, in this case, the PMI analysis node needs actually the input file of the compound information, but I just didn't put it there. That's why it's red. And this one, I didn't select the proper input file of the FASTA. That's why it's still red. And when the KNIME is working, it's always like this. So the process is now in this position. So it's also very easy to see where the programming is going. And the last thing is it's also possible to connect many applications within the KNIME. Like all applications shown here can be connected within the KNIME. So it's so important actually. For example, original compound information could be stored in AWS, and the biological data can be stored in Oracle, and the combined data can be shared in Google Drive with restricted people. This type of the system could be built up just through the KNIME, and it automatically updated like every day or every week or whatever is fine. So that I just inside the KNIME is so useful. But the next question is, how can people learn the KNIME? Well, please Google the word KNIME Community Hub. It's shown here, like KNIME Community Hub. So this is a platform where many KNIME users upload their own workflows. You can simply download the workflow and put your own data as an input file. In my experience, the easiest way to run the KNIME is to mimic kind of existing workflows. And if you got error that you cannot solve, then please share your problem in KNIME community forum. Here, forum. The many KNIME users there will give you the advice for free. And also, I highly recommend joining the Just KNIME Challenge. It's a weekly event hosted by the KNIME team where they give you a small task to solve using KNIME. And then you need to share your own solution, your own workflow in KNIME Community Hub. An example of the answer will be shared after all. So it's very great opportunity to run the KNIME, like even for me. Oh, that was the last slide.
That was the slide? Cool.
Yeah.
That comes in handy because actually I have a lot of questions. Just to make sure, like everything you showed today is basically open source, right? Maybe the ChemDraw, that's a commercial bit, but
Yeah.
all the KNIME nodes, like all the workflows, anybody can reproduce that once you have applicable data and you have your molecules, right?
Yeah, I have never paid. Yeah.
Cool. Do you have a tip? Like you covered these molecular structures, the SDF, the mol. If you're really a complete newbie, where can I get examples? Are there any public databases or something where I can actually just download something so I have the data to start playing around with it in KNIME?
Yeah, I feel like just access the KNIME Community Hub forum.
Okay.
And then just search the word chemistry or library or substructure or something like that. And then downloading the example like which somebody made already, and use your own input file. That's the easiest way.
Okay, cool. So I assume there will be some data in some of the workflows. Maybe a little tip from me, if you want to get started with the molecular formats and everything, I always go to Campbell. It's my kind of go-to library. They are also for free.
Yeah, Campbell
and what another question, because we were talking obviously and preparing for this webinar, and you also told me that you can do Python. So you're actually not one of these kind of locked web bench scientists, and you could do all of the stuff in Python. Like, what's still the argument for you to do everything or like a lot of stuff in KNIME?
I mean, I like Python too, but KNIME is kind of easy to do something already. I mean, it's not so complicated. I don't have to spend a lot of time to do like easy things, and they just drag and drop and making their useful workflow very easily. So
Okay.
I often use the KNIME from the beginning and at the first, and then if we have to combine with something, then I just write a Python.
Okay, so most of the functionality you need is actually available as KNIME nodes, and just sometimes you have to deviate to Python. Did I get that correctly? Okay.
Yeah. Like especially something from journal.
Cool. Actually, we do have a question. Can I generate molecules using smiles with RDKit molecule?
You mean, yeah, yes?
I mean, yeah, can we generate a molecule with RDKit? It's a bit of a generic one.
Like making the partial library or something. Maybe.
Yeah, I'm also puzzled. Maybe that person can specify again, like, what do you mean, kind of like the sketching part? I think for that one, this is not the RDKit bit. For that, you need a molecular sketcher. So as Hiroki showed, ChemDraw. But we're also having a free one that is using Catcher under the hood. So that's another library. And let me check if there are other questions from the audience. Is there any open source node for running QSAR in KNIME? I'm not exactly sure if that's your, like, if you do that actually in the lab and if it's part of your work, but do you have some expertise there?
Yeah, but I mean, there is some like papers talking about the QSARs using the KNIME. So you can just download the workflow from that paper too. But the number of their kind of model are not a lot. But it's also kind of easy to develop your own model in the KNIME. Like machine learning is also very useful in KNIME too. I mean, it's visible, so you can kind of see what you're doing. So it's also kind of a good chance to learn what machine learning is.
Yeah, I'm also thinking, but I think we do not have a node right off the box that would kind of do that. There was a library around, I think from Cody or so, that could integrate it into KNIME, but I would need to check if that is still being maintained. And we do have another question. Can we input wet lab data? I don't know if that's part of your work too. For example, activity for structure-activity relationship analysis into KNIME. Do you do that in your everyday work, that you're actually having assay data and you kind of correlate that to, are you doing this kind of analysis, or you're really just more on the pure chemistry bit?
Well, it's possible. I mean, I'm doing that way too. Like I got the wet data from the experiment and put it in the kind of Excel or KNIME and combine everything together. Like in drug discovery, we have to collaborate with kind of many people, like biology, chemistry, and toxicology, and everything. But all data cannot be stored in just one folder. So we have to we have to go to many kind of website, like the same as the one I introduced last thing. So AWS, Google Drive or something, we can just combine them together and make it kind of visible. So you can just make a list of a compound with many properties, like many wet data.
Yeah, actually I had a use case that's really 4 or 5 years ago where I also used some old data from my lab days, and I built a KNIME workflow to kind of analyze it, restructure it, just the assay readout, map that one with molecules. It's also still somewhere on the hub. Actually, we get a lot of questions. There's another one. That's also a really good one. Can we make custom KNIME nodes? I can already say yes, you can. So it's since 1 or 2 years, it's possible that you create these KNIME nodes in Python. Before it was pure Java. Maybe question for you, Hiroki. Have you done it, or are you considering doing it?
I'm considering doing it, but unfortunately, the node is already enough here, so I didn't make it by my own. So, yeah, I mean, if I have to make it, I just rely on Python, so I don't make it.
Yeah. Okay. So you just integrate a Python snippet code to pull in or execute it in Python. You're not creating a whole extension on your own, and yeah, that is also working. Let me check what else we have around.
I mean, maybe I can just share some disadvantage on mine. So that it needs a lot of memory. So that's a problem. Yeah. So if you want to handle like a large data, then KNIME cannot kind of solve it. It's very easy to visualize. It's easy to kind of think about the workflow a lot, but the memory is a problem.
That's yeah, we're encountering that ourselves every now and then. I feel you there. Don't believe me. Yeah. We have another question. How does KNIME work for me as a method to prepare a unique assay as to medicines against these toxins? I'm not sure if we get there completely, but not for KNIME, like you just can do the data analysis with it. So the whole assay design, like that's still on you. But once you get a readout from whatever machine you're using, a plate reader or something, and you want to restructure your data, bring it in a nice machine readable format, match that with the molecules you tested or the compounds, stuff like that. So as soon as it comes to the data analysis, I think then you're really good with KNIME, but I'm not sure if we can really help with the preparation for that. Yeah, not.
I mean, KNIME cannot build up the kind of a protocol of experiment, so KNIME can just analyze the data.
Unfortunately not. Yeah, and
but it's also important to make a viewer. Like you do a lot of assays and you collect many data, but I'm sure that if you have over 10 types of data, then you will kind of lost where you are, and this makes, I mean, the KNIME can solve that problem. You can just make a viewer for everything. Yeah, it's also easy.
Yeah, that's these interactive views that you had, right? Kind of this component that helps them with yeah, just evaluating the results of your assay.
Yeah.
I would have maybe one more question. You showed this dashboard for, I think it was called compound registry or something, and you had the CAS numbers in there. And I remember, and it's also for a lot of our customers, it's a huge hassle to get from a CAS number to these molecular structures that are actually that KNIME can deal with, because with the CAS numbers, there's not a lot of cheminformatics you can do. Do you have any tips there? How do you convert that? If you get a CAS number, how do you get the molecular? Yeah, exactly that slide. How do you get the molecular structure from that one?
So I'm sorry, I'm not hiding it, but in this component here, it has an API, and I think that it's also possible to use kind of a KNIME node to get their information using the API. But in this case, I feel like I use a Python here. Sorry.
Okay.
It's also possible by
yeah. And maybe a little bit of an explanation for the ones who don't know what an API is. It's basically letting the computers talk to each other instead of us going to a browser, for example, to Campbell or something, look for the name of a molecule, and then they get the molecular structure. You can basically let the computer do that. Which API are you using there? Is it the CAS one or ChemDraw, or? And I assume these are commercial offerings, but maybe good to know.
Well, maybe I have to open it because it was built like 5 years ago, and I forgot the inside actually. It's working without any problem so far, so I just didn't change it.
Okay, cool. Let me also check if we have any other questions. Can KNIME use files from GraphPad Prism? Oh yeah. I think the short answer is still no. I investigated that quite a time ago because I also use GraphPad Prism in my PhD. So for me, that was the go-to analysis tool. And to my knowledge, we haven't gotten yet to kind of decode their files and their format, so we can actually import it into KNIME. But also I have colleagues on the chat, not only Greg and Steve. So maybe if you have more input on that and there's some news, please go ahead and answer that one. And I think with that, we are quite good on the chat. If there are no more questions, thanks so much, Hiroki, for joining us. That was really a lot of fun. I hope that was also useful for the audience. I had fun. We had fun. And also, let me show you some more wrap-up. So if you found it interesting and maybe you're following this whole KNIME 101 webinar, please join us for the next one. So there we're going to cover how to generate PDF reports with KNIME. That's also going in the direction of these components that we were covering today. And also maybe more interesting is we're running a course, Introduction to Working with Chemical Data. So that's run by me and my colleague Torol. It's happening in June, so I think 2 weeks from here. And we're also not as much as Hiroki, like covering the different molecule formats, but go a little bit more into the chemistry direction. So we're exploring chemical data. We're going to do an R group decomposition. We investigate clustering and also similarity search. So if that's of interest for you, I would be super happy to see you there in 2 weeks. And also that's it. Thanks everyone for joining. If you want to stay in touch, obviously KNIME.com. Also find us on LinkedIn, where we post a lot of stuff. So if you want to keep updated, like what's going on within the company. I also want to highlight the KNIME TV. So we have a lot of resources, a lot of educational videos, how to do things. If you're ever stuck, I personally do that even. Go to our YouTube, find some video that help you. And yeah, with that, I think we're good to go. Thanks everyone for joining, and hopefully see you soon somewhere. Have a nice day.